Hello guys, I am Shubham from Shubh Tutorials. This video is about nucleotide excision repair. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. We are discussing the repair pathways. In the previous video, we saw base excision repair. So nucleotide excision repair is also similar to the base excision repair. But what is the speciality? It does not recognize any specific lesion. Okay, it does not recognize any particular lesion. Does not recognize any particular lesion, means damage. It cannot recognize particular damage. Okay, what it recognizes? It recognizes distortion in the DNA. Okay, it recognizes distortion. Distortion means change in structure of the DNA because of damage. Okay, so it recognizes the distortion in the DNA and then it starts repairing the damage. Okay, based on the distortion, structural changes in the DNA. So let's see the processes. Firstly, let's see in the prokaryotes. So this is in prokaryotes. In prokaryotes, proteins which UVR proteins. Okay, the order is A, B, C, D. Remember like this only, A, B, C, D. So first comes the protein UVRAB. UVRAB as a dimer. Okay, two molecules of AB are there which are bind to each other, right? These are the dimers and this will come and recognize the distortion. Okay, distortion. See, this is a distortion, change in the DNA structure, right? Then what happens, as the distortion is recognized, A will be released out. A will be released out. A is important protein for the recognition of the distortion. Okay, as we are seeing, they are working in the dimer, but as the distortion is recognized, A will be released out. Means what we can understand, A is important. A's function is to recognize the distortion, or we can say detect the distortion. Then the B protein, UVRB protein, are melting the DNA. We can see a structure from change in the structure is seen from double strand to like this. So this is called a melting. Okay, so this is done by UVRB, right? So we can say UVRB is a type of a helicase, right? See, we can see here melting. See, bending, we can see a bending of DNA in this picture. Then what happens after melting? Next protein will come C. Okay, UBRC. This will come. This is a nuclease. Okay, this is a nuclease making incisions both the ends, 5' and 3'. See, we can see arrow here. This will make incisions. So making of incision is also done very specific. Means 5'. Let's draw DNA. Okay, now here is the lesion, damaged nucleotide. Okay, damaged nucleotide. So this is the 5'. This is the 3'. 5' cut. 5' cut is made 8 nucleotides away. 8 nucleotides away. Okay, from the lesion. And 3' cut is made 4 to 5 nucleotides away. So specific cuts are made. Right? 3' cut is 4 to 5 nucleotides away from the lesion, and 5 prime cut is the 8 nucleotides away from the lesion. Alright, and the next protein. Now the cut has been made, but DNA is still attached, okay, to each other, right? So the next protein will come, next enzyme, UVRD. Okay, we can see next enzyme will come UVRD. UVRD is a helicase. See, we can see UVRD is a helicase. It will take away the strand out. Okay, take away the strand out or remove the strand. In the previous step, we saw UVRC is just making the cuts. Okay, these are the cuts. UVRC will make a cut, right? But the DNA, the nucleotides are still attached to each other. So the next step, UVRD helicase, will come and break the bonds and separate the damaged strand. The damaged strand or the strand which was cut will be removed out. We can see. See, this is the strand which is removed out along with UVRD helicase, as well as C is there here. Okay, so all these are removed out. The gap, then there will be gap. The gap will be filled by DNA polymerase and the ligase. Right? DNA polymerase, basically repair enzyme, DNA polymerase 1 in prokaryotes. So 12 to 13 nucleotide strand is removed. Okay. So this gap then will be filled by DNA polymerase 1 in prokaryotes, and the last seal which is remain is filled by the DNA ligase. Okay, so this is in prokaryotes. These steps are very easy to remember in the order UVR A B C D. Right? Now let's see the process in eukaryotes. In eukaryotes, so same processes, nothing different. Here the first protein comes XPC, which recognize the distortion. Okay, recognize distortion, similar to UVRA. Then the next proteins, DNA melting, right? XPA and XPD. DNA melting, second step. So these are similar to UVRB. Okay. Then the nucleases, cutting the DNA. Cutting, or we can say neat. Right. So those are nuclease. For 5 prime, two types are there, for 5 prime and for 3 prime. That is ERCC1 XPF. XPF. Okay, for 5 prime cut, and for 3 prime cut, XPG. These are the proteins, and then the gap will be filled by DNA polymerase beta and DNA ligase. Okay, one more protein after DNA melting here is the RPA. We have already discussed about RPA. It is a single-stranded binding protein, right? So that the melted DNA cannot get bound to each other again, right? So prevent rebinding, RPA works. Okay, single-stranded DNA binding protein. So this is in eukaryotes. Now These are basically XP proteins. XP proteins. Okay. So what is XP? XP is a disorder caused in humans. Xeroderma pigmentosum. Okay, Xeroderma pigmentosum. As well as this is XP. As well as UVR proteins. UVR proteins. What they do basically, see, this is UVR. UVR stands for ultraviolet radiation. So damage that is caused because of ultraviolet radiation is repaired by these proteins. Okay, we can see thymine dimer is repaired by these UVR proteins. Damage that is caused by the ultraviolet radiation. So this dimer, as well as this is in eukaryotes, then this disease is also caused in humans where there is mutation in XP protein. There is mutation in the XP proteins. So the repair is not done. Okay, the damage that is caused due to UV rays, the repair is not done properly and it results in skin problems. Okay, so this is basically a skin disorder. It can also result in skin cancers, right? As well as this is a autosomal recessive condition. Autosomal recessive condition. Okay, pattern of inheritance, right? So this is all about nucleotide excision repair. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.